Well, we've begun talking about proteins, which now you know are like my favorite biomolecules. And we've learned that proteins and peptides, by the way, are polymers that are built from monomers that are called amino acids. Um, let's see. Let's use my pen this time just to shake things up. Okay, amino acids are the monomers. And here we can see 1, 2, 3, 4 amino acids. Each 1, 2, 3, 4 amino acid. Each amino acid has got one amino group, one carboxyl group, a little hydrogen over there. And then what makes the 20 different amino acids that are used to make human and life on planet Earth's proteins, what makes them different from each other are these side groups. And I told you at the last video that the amino group on one side is attached to the carboxyl group of the adjacent amino acid, and the carboxyl group is attached to the adjacent amino acid, just like this, right? So they do go together kind of like beads on a string, but what is unique are these side groups. So I want you to imagine for a moment that I have put together a string of beads, and let's make it a necklace, and let's make hanging off of it little dewbobs, okay? And it turns out that each one of those is a magnet. I don't know if you know, but magnets have got a positive side and a negative side. And when the positive sides get close to each other, they push each other away. If a positive gets close to a negative, click, right? Well, let's imagine that these 4 amino acids are going to be those charged side groups that we looked at. See, remember here, 21 amino acids, 20 for life here on planet Earth. Remember these guys, that they're electrically charged? Let's imagine that I'm going to put 4 positively charged ones right next to each other. Okay, here's my little necklace. Things are hanging down, and I've got 4 positively charged ones. What would happen? They would try to push away from each other. So when they tried to push away from each other, they would kink or whatever the necklace. You could try to straighten it out, but as soon as you let go, they'd push away from each other and they would change the shape of the necklace. That's what amino acids do when you build a protein. Let's imagine if instead of that, let's imagine this one's positively charged, this one's negatively charged, this one's positively charged, and this one's negatively charged. Let's just imagine that. Okay, now what would happen? Well, now they would go click, click, click. And so whatever you have, they go click, 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 they click together, and then they would form, again, a little bend in the necklace that you're building. And you could separate them out and go, okay, let's separate them out, but they'd go click, 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 right back together again, right? So it is the interaction, sometimes ionic, sometimes hydrophobic, hydrophilic, sometimes other things, the interactions between these side groups that end up as the beaded necklace is being made and the beads are coming out that side, they will curl themselves into specific shapes. Okay, so let's go back to here. There are 20 amino acids. There are some that are charged, some that are polar, but uncharged. These all dissolve really well in water. The ones that don't dissolve well in water, there are quite a bit of them. And when they don't dissolve well in water, then they also will cause that little necklace to curve because they'll all try to keep their non-polar side groups pointed towards each other to keep them away from the water, to keep their little side groups dry, right? So proteins build their own structure. Now, this is what you need to know about protein structure for right now, and we're going to connect it to what you're learning in lab number 2. First of all, when protein biochemists study protein structure, the reason we're studying protein structure is to try to predict protein function. And we're still at the point where there are many proteins in the human body. We don't know what they do, right? And here's how we approach that problem. First of all, there is what is known as primary structure. I told you that proteins are made like beads on a necklace, and the primary structure is just what is the sequence of the amino acids. For example, bead number 1, isoleucine, bead number 2, glutamine, bead number 3, proline, bead number 4, I'm going to put another isoleucine, bead number 5, threonine, right? Boom, 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 boom. Right? Keep in mind that let's do the situation where it was supposed to be positive, negative, positive, negative. Okay. Let's imagine I'm supposed to have positive histidine, negative aspartic acid, and then an arginine, and then another aspartic acid. Let's imagine that was the sequence it was supposed to be. If I did that, then they would have all go click, 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 and attach to each other, right? But what if I made a mistake? What if instead of I'm going to start with the histidine, but the next one was supposed to be an aspartic acid, but I made a mistake and put a lysine instead, and then an arginine, and then an aspartic acid. Okay, what if I did that? Well, you wouldn't have they wouldn't have clicked together, right? I put 3 positively charged ones in a row. So where they were supposed to click together and form a stable little structure, now they're busy pushing themselves apart. Now, if it comes to a necklace, that just makes a necklace that looks differently. However, that's not what proteins are. Remember I told you proteins are machines, and let's think of proteins as a robot, okay? Let's imagine my protein robot was supposed to have little elbows here like this, and he was going to grab things and put them together, and grab 2 things and put them together, and grab 2 things and put them together. All right. And let's imagine that the positive, negative, that click, 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 that's what made the bend in my elbow. All right. And so when it's bent the way it's supposed to be bent, my robot can do its job. Okay. But if you made a mistake and you put the wrong amino acid there, now instead of my elbow being bent like this, it's kind of broken. And so now I can try to move the way I was moving, but I can't grab the things and put them together anymore, right? It doesn't surprise you that the shape of a robot is what allows it to do its job, right? If a robot's got his elbows bent right, it's got its hands shaped in the right shape of their hands, right? That they can go ahead and do their job, and that if you bent the elbow, if you put it together wrong, it wouldn't do its job. When we say that there is a gene mutation that's causing a disorder in a person, the reason that there's a gene mutation is there's a mistake in the instructions for putting together a protein. That's what a mutation is. It's a mistake in the instructions for putting together a protein. So a mutation might be instead of histidine, aspartic acid, arginine, aspartic acid, it was histidine, lysine, arginine, aspartic acid. That would be a mutation, and it would ruin the shape of it because it changed the primary structure, right? So levels of structure in a protein. First, we just got to think, do we have all the amino acids in the proper order? Primary structure, right? Now, okay, just for a second, let's go ahead and jump down to tertiary, okay? Tertiary means 3, right? And so I think of tertiary structure as its overall 3-dimensional shape. And in order to have tertiary structure, that means when I'm done, do I have a robot that grabs things and smashes them together? Or do I have a robot that grabs one thing and rips it apart? Or do I have a robot that just attaches to other little things to make some giant structure? Or, overall, okay, when I'm built, what have I made in terms of that one polypeptide? Okay, right. So primary, tertiary. Now, I skipped over secondary because people get confused with secondary, but you won't get confused with secondary. Why? Because primary, that was the order of the amino acids. Tertiary is, hey, what do I have? Do I have a robot? What do I have, right? What is secondary? Secondary are the individual pieces of the protein, right? So if I had a robot that could travel 60 miles an hour down the freeway handing out tickets to people who are driving with one person in the carpool lane or something like that, then what would be the pieces of that robot? We probably need wheels, right? And we would need some kind of a ticket taking machine, and we'd need a siren. Okay, that would be my robot. So overall, tertiary structure is I have got a robot that gives tickets on the freeway. It's made out of, metal, glass, blah, 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 blah. And the parts of it, it is going to have wheels and a ticket printing machine and a siren. Okay. Now, as a protein biochemist, you will know something about a protein by knowing that it's got wheels and a siren, right? Because if it was a copier sitting in someone's office, it doesn't need wheels and it doesn't need a siren at all, right? Now, it might have something that looks like it's a ticket giving machine, but that's just the part that's printing out documents, okay? So primary and tertiary, we start with those. Secondary is the in-between part, right? Now, what is quaternary? Not all proteins have got quaternary structure. Not all of them do. Only really large proteins that have got many individual protein subunits put together have quaternary structure. I'll give you an example of quaternary structure, hair, okay? Hair and fingernails, they are made out of protein. I'll tell you honestly that an individual protein, its tertiary structure, way too small to be seen even with the most powerful light microscope. You cannot see it, way too small, okay? And yet it's easy to see my hair. It's easy to see my fingernails. Why can I see hair or fingernails if they're made out of protein? Ha! Because there's enormous quaternary structure in those proteins, billions and billions in that situation. I think the situation in your textbook is hemoglobin, and I wish they would stop using hemoglobin as an example of quaternary structure, because hemoglobin is made out of 4 proteins, subunits. And so I think it makes people misunderstand that quaternary structure doesn't mean it's got, when you put 4 proteins together, that's just a coincidence, okay? We're going to be talking about the quaternary structure of myosin and actin when we get to studying how muscles contract. They've got huge quaternary structure, okay? So protein conformation and denaturation. I usually say denaturing, though, okay? So the conformation, that's the word for the 3-dimensional shape. If I say a protein has got its normal or its native conformation, I'm saying it's got its normal shape. And I told you this before, that the shape of a protein determines its function. If the shape is wrong, it won't be able to do its job. That's not surprising. By the way, most proteins, even the somewhat boring ones, move a bit as they do their jobs. So they really are like robots, okay? Now, denaturing. Denaturing is going to be part of your lab this week 2 in your lab, and it is an irreversible disruption of the protein shape, right? So there are things that can happen that will damage the shape of a protein. If I took your lovely Xerox machine robot and I put it through a fire and it melted, and I gave it back to you and I said, here's your machine back, you'd go, well, it's not going to work. And I'd say, how do you know? It's like it's melted. That would be like denaturing. If you heat up a protein really high, that is a most reliable way to denature a protein. And once it's denatured, it cannot do its job and it's not coming back from that. You also can denature a protein by putting it at a pH that it does not like, all right? Now, proteins, enzymes that work inside of a living cell, they usually like a pH that's relatively close to a neutral pH. However, there are plenty of enzymes that like a wacky pH, like a superacid pH of 2. However, we can generalize that if you take any protein and put it into a pH that is not normal for it, that's a good way to denature the protein and wreck it. Radiation can wreck proteins. Chemicals can wreck proteins. If you take a raw egg, the egg white is clear and it's kind of a jelly, kind of liquid kind of thing. If you cook it, it'll turn white and rubbery. If you put it in the refrigerator, it doesn't go back to being clear and liquid, right? And the reason is that the protein that's in the egg white is called albumin, and you have denatured it, and it ain't coming back from that. Ready? Oh, okay, I can't finish without showing you a couple of my favorite proteins, okay? So, proteins. This is a cartoon of a protein, but it's very much this one. It's very much shaped what it's like. So this part right here, I'm going green. Okay, this part right here, okay, that's kind of like the head of the protein, and that head is holding on to a big balloon, and this big balloon is filled with more proteins. Why is it carrying it? Because that big balloon filled with proteins is going from where it was made to the outside of the cell. How is that big balloon of proteins going to get there? This protein called dynein, you don't have to know its name, is going to walk it there, all right? And these are its two long legs, and these are its big fat feet, and it's going to walk one step at a time, carrying this enormous balloon of protein on its head, and it's going to carry it kind of like a really slow mailman from one part of the cell to the surface of the cell. That's what proteins do. See what I said? They're like robots, right? Now let's look at this one. I told you that protein biochemists care about that secondary structure, the little parts. By the way, the most common parts of secondary structure is the alpha helix and the beta. Sorry, I was going to do the Greek letter beta, but why do you know the Greek letter beta? The Greek letter beta pleated sheet, okay? Those are good examples of secondary structure. But look at this one. Do you see how it looks like a tunnel? It is. It's a tunnel. This is a special secret tunnel through the phospholipid bilayer. We're going to talk about how the phospholipid bilayer, one of its jobs is to not let stuff go from the inside to the outside or vice versa. So when anything wants to go in or out of the cell, some things need their own special tunnel like this protein. So if I were, for example, glucose, I would bind right there. And when I bound right there, that would cause this machine to go chunk, chunk, chunk. And that would cause it to open up so that I could walk on through. Yeah, proteins are cool. Proteins are very exciting. Ready? And you will be studying enzymes. And we will pick up here at the beginning of the next lecture.